So I'm delighted to be here today and tell you a little bit about some modeling approaches for trying to link some of these imaging modalities and some of the network modalities that we've been talking about today. My talk's broadly going to follow this outline. I'll start with a network science framework for representing structural and functional connectivity. I'll tell you a little bit about that. I'll present some empirical evidence that these two network modalities should be linked to one another and are in fact linked. And I'll get into the guts of the talk at this point, describing several modeling approaches for trying to link those two imaging modalities, those two network modalities, focusing on two distinct sub-approaches, one using biophysical models, and the other using what we'll call communication dynamics, which are really stylized models. And then I'll end with some challenges and frontiers for trying to link these two different kinds of modalities. So I'm a network scientist. I start from the perspective that the human brain is a complex network, and that's the modality that we should be studying it at. These networks are fundamentally multimodal, so we've already heard a lot about diffusion imaging and tractography for generating structural networks. We've heard a lot about using resting fMRI to generate functional networks, and I'm not going to spend a lot of time talking about the processing strategies. I'm going to play a game where we assume that, as challenging as all of that processing may be, let's pretend like we have it solved. It's not, of course. Let's just pretend, for the sake of this modeling exercise, that we've solved all of the processing issues associated with generating structural and functional connectivity networks. So I'll show you these two side by side. We can represent them as matrices. So on the left is a structural connectivity matrix. The rows and columns correspond to brain regions. The colors indicate the presence or absence of a structural connection, so a fiber tract linking regions, and the strength of that tract. And on the right-hand side is functional connectivity. It's just the correlation structure of resting bold data. And when you look at them side by side, there's a passing resemblance. They're not indistinct from one another, but that relationship is what we're trying to make a little more solid today. We're trying to make that relationship a little more explicit. And of course, these two network modalities, they have some common features. They can be represented as networks. The brain regions are nodes. The structural connections are edges. The correlations can be represented as edges. They have multi-scale organization. We can interrogate their structure at the level of individual regions, at the level of the network as a whole. Intermediate scales, where we look at clusters or communities. But they also have many important differences, and some of these we've already discussed. Structural connectivity is fundamentally physical. Like if you crack open a brain, in principle, you could see the white matter fiber tracts to some extent over maybe a short enough time scale and with the appropriate spatial scale. The number of structural connections may be finite and innumerable. Those connections have complex biophysical attributes, and they tend to change slowly in time. And the structural connectivity kind of represents a backbone. It's the structure, it's the paths, it's the roads that shape signaling between different brain areas. It shapes the flow of activity of the brain. Functional connectivity, on the other hand, is fundamentally statistical in nature. It's always estimated as a correlation or a partial correlation or some kind of statistical relationship between activity. In principle, there's no constraint on the number of configurations that a functional network could adopt. Those connections have their own attributes, which may or may not be of interest, and they change rapidly. And fundamentally, functional connectivity represents the outcome of those signaling processes constrained by the structural connectivity. And collectively, we call the study of these two modalities connectomics. And the relationship that we posit is as follows, and I've already kind of hinted at this, but structural connectivity, the white matter pathways, represents the highways of the brain. It represents the conduits along which signaling occurs. That signaling, that communication between brain areas is taking place along structure, and the net effect of these communication processes is correlated activity, which we measure with fMRI. So there's a structural connectivity serving as a stable backbone that structures the flow of activity, the flow of communication, and the end result is correlated brain activity. And there's lots of really nice evidence that functional connectivity should have this anatomical or structural basis. Much of it's correlative. So there's a lot of examples where strong correlation of spontaneous activity is underpinned by strong anatomical connectivity. This is in macaque, but you can also do the same thing at the whole brain level with human data generated from fMRI and diffusion imaging. But there's also some causal evidence too. So this is a nice study from Jill O'Reilly where they start with an intact macaque brain. So this is the functional connectivity matrix. And then they're going to clip a bunch of callosal fibers. So pay attention to these interhemispheric correlations. This is before cutting these callosal fibers. There's an acute insult to the network, and suddenly interhemispheric coordination goes away. So there's a strong structural basis for the correlation structure that we observe, and probably in a causal way. This is I think a good example of that. So how do we model this? How do we go about modeling structure-function relationships? There are many ways, but I'm going to focus on one, and it works like this. So here's our structural connectivity matrix. We get it from maybe diffusion MRI plus tractography. And the bottom is the functional connectivity matrix, the set of all the correlated bold activity. One way we can try to model this relationship is using forward models. So we take our structural connectivity and we ask, what is the dynamical process that, when we constrain it by structure, gives us something that looks like function? So we're trying to build a dynamical model constrained by the anatomy that gives rise to similar correlation structure, right? So it would look something like this. We have our forward model structure, which is constraining some dynamics, and it gives rise to some correlation structure for the model. We can then try to compare this to the observed correlation structure and ultimately choose our model on the basis of whichever model matches the two, creates the greatest correspondence. This is the approach that I'll focus on today, this sort of forward modeling using structural connectivity plus some dynamics to give rise to correlation that looks like the observed correlated activity. You could, of course, try the inverse problem, asking if I know the correlation structure, what was the underlying anatomy? It's a much more difficult problem. It's ill-posed, and it's not something I want to spend a lot of time talking about today. In the connectomics literature, there's actually not a lot of people studying the inverse problem, but there are a lot of approaches for taking structure plus dynamics to get simulated functional connectivity. So what kind of models do we use in this forward modeling approach? Well, there are in fact a spectrum of models, ranging from simple statistical models that seek only explanatory relationships to those that are biophysical and ideally closer to mechanism. So a good example of the biophysical models are neural mass models. So the neural mass model assumes that there is a population of excitatory and inhibitory neurons at each brain area, at each node in the network. They live there, they talk to one another, and they can excite themselves or inhibit themselves. And these populations are then coupled to one another via the anatomical structural connectivity. And we have very detailed differential equations that describe at the population level how voltages and membrane potentials should change. These have parameters like related to channel conductance and concentration of ions. It's very close to the neuroanatomy. It's very close to the biophysics. And then you could, in principle, just evolve the system over time, integrating many coupled differential equations. On the other hand, there are the simple statistical and explanatory methods for trying to relate structure to function. Canonical correlation analysis is a good example. Partial least squares is a good example, where you're simply taking your structural connectivity and functional connectivity and trying to use some statistical model. There's no dynamics, but using some statistical model to generate some correspondence between the two modalities. Situated between these two extremes are a series of other dynamical models that gradually lose their biophysical reality, ranging from simple dynamical models based on diffusion processes, oscillator models, and then the communication dynamics, which we'll talk about later. But this is not a full representation of what is available. There are many abiological dynamical models that people have and continue to use to try to relate structure to function. But we'll focus today on the biophysical and mechanistic, and then ultimately the communication dynamics. So how do these biophysical models work? Well, in a little more detail. So this is the binary macaque matrix. We saw part of this earlier when Dr. Van Essen presented it. But here, the white cells indicate the absence of a reconstructed tract between projections between areas. Black indicates the presence. And we're going to take this connectivity matrix and couple it with those biophysical models, the neural mass models, which we already saw those differential equations. When we simulate this dynamical system, it gives rise to population level activity. We can convolve that with a bold-like response, and it gives us simulated bold correlation, simulated functional connectivity. Why is this useful? Well, on one hand, we can simulate the effects of perturbations or lesions to the network, and by removing individual edges or by removing nodes and all of their connections from this matrix. Run the model again, and you'll get a simulated bold correlation corresponding to the case where that brain region doesn't exist. Maybe it was ablated by surgery, you could imagine. This framework also allows us to simulate the effects of inputs. By adding them, injecting them into these dynamical systems, you can add input to the system and see how it evolves, and it'll, in principle, change the simulated bold structure. But the open question, of course, is this simulated data related at all to the observed functional connectivity? Do they look alike? So when they first started using these neural mass models, there wasn't a lot of real data. There's some connectivity matrices, but not a lot of real data to compare the simulated correlation structure to. Well, finally, they got a hold of some actual fMRI data from macaque. This was in 2012. And on the left-hand side, you're seeing the observed functional connectivity from that fMRI data, resting fMRI data with macaque. And then on the right, you see what the model predicts. So if you squint with the right eyes, there's a correspondence. It is imperfect. And we can assess that relationship statistically, and sure enough, they're correlated. It's, again, not a perfect relationship. At the end of the day, it's a very stylized model still, and missing lots of important neurobiological detail. But it does a reasonable job. The biophysics, there are no free parameters. Most of the free parameters are set to biophysically realistic values. We can get those from data. So this is without any tinkering, without any model fitting, essentially. Just running the model, coupling it according to structural connectivity, and you get something back that doesn't look so bad. So there's some partial agreement. The next step, of course, is to see whether this same approach works for human data. So this is human data collected or reconstructed from diffusion imaging and tractography. There is some observed functional connectivity from those same subjects, and the model generates some simulations. So taking the structural connectivity, using that to couple and to constrain a series of differential equations. And of course, there's a relationship between the real structural connectivity and the functional connectivity, the simulated and the real. And then the simulated functional connectivity looks a lot like the observed. So that's quite nice, right? There's some correspondence now. There's some predictive capacity using this very complicated neural mass model that preserves something about biophysics that we think we like. So it's good. It incorporates some biophysically plausible mechanisms. But on the other hand, it's really computationally intensive. I don't know if you've ever worked with tens of thousands or thousands of couple of differential equations. Even if you have access to high performance computing, it takes a long time to generate these simulations. At the time some of these first studies were carried out, generating 10 minutes worth of simulated data took like 7 or 8 hours. It's faster now because that was 2007 or 2009, but it's still a computationally intensive process. And consequently, it's not like you can systematically assess the effect of removing a node or removing every connection. It would take a long time to assess the contributions made by those smaller elements in the network. And at the end of the day, it also only has quite modest predictive capacity. So in the previous slides with the macaque data, the correlation of the observed and simulated functional connectivity was like 0.5, and with the human whole brain it was about the same. So it's quite modest. So there's some reasons that as biophysically plausible as this may be, we may not be happy with the biophysical approach. And this has motivated us to start looking at what we call communication dynamics. So in communication dynamics, rather than studying something that's biophysically plausible, we abstract away a lot of detail and study stylized processes of how brains could, in principle, communicate. These approaches end up being computationally tractable, eminently so, and as a consequence, it's very easy to assess the rules of individual areas, individual connections, and they tend to have greater predictive capacity in the sense that they are much better at predicting the observed functional connectivity using these simple models. So I'll give you some examples, but I would encourage you to read the Nature Reviews Neuroscience paper that came out, I guess now last year, that walks through a lot of what I'm presenting now. So communication dynamics is some set of stylized processes, some policies by which brains can, different areas in the brain could talk to one another or exchange information. So here is our structural connectome. Imagine there's some source node in the brain, some place where some signal is emanating, and there's a target. These two areas maybe want to talk to one another. How do they take advantage of this structure to pass signals, to pass messages? There's a lot of ways that we could imagine this happening. One, maybe they just communicate along shortest paths. So the path through the network that involves the fewest number of steps. There's a nice network science-y feel to it, a nice graph theoretic feel to it. So that is an example of a communication dynamics process from our perspective. And how this fits into our modeling approach. So before we were trying to take structure, and these should be reversed actually, to take the structure and make predictions about function. Let's replace the structure with some matrix, in this case the shortest paths matrix, that summarizes the communication dynamics and ask, can we use this new measure of communication to make predictions about functional connectivity? So it's just substituting one for the other. We could also consider other processes like instead of using the shortest path, what if you had a random walker with no knowledge about the network, just moving over from one node to another, hopping along outgoing connections. We can try to summarize that process with something like a mean first passage time matrix. So how many steps on average would it take for a node starting at this source to get to the target, for instance, and then build our models on the basis of these stylized kinds of communication processes. So we're sacrificing this biophysical realism for the ease of interpretation, essentially. And we've done a lot of work in this space, and I'll give you one good example. We developed a metric called search information, which tells us how hidden a path is in the network. I'll give you an example, and I'll try to make it relevant to conferences, right? So I can guess that some of you are going to go to a bar tonight, or maybe a pub, or maybe a brewery, or something like that. How easy is it going to be after a couple hours there to get back to your hotel? How hidden is that path from the bar to your hotel? It's a question, right? If the road was a straight shot with no intersections, doesn't matter how much you drink, just keep walking forward and you'll eventually get back to your hotel. That's a very, very obvious path. It's not hidden at all, right? On the other hand, if you had a path or if that road had many intersections and you had many choices to make, do I go left, right, straight? Another intersection, left, right, straight? Maybe you end up somewhere unfamiliar, right? So this is an example of a hidden path or a path that's very hidden versus one that is not. And we quantified this in terms of an information theoretic quantity, and we use this to make predictions about functional connectivity. So here's the predicted functional connectivity, and on the right is the observed. So it does marginally better. And it's based, again, on a very stylized communication process. And you can see some examples of some seed correlations for this process. Recently, we used the same measure to demonstrate that it's not that the predictive capacity of search information isn't limited to fMRI data. We can predict functional connectivity from intracranial recordings too. So we have a whole brain set of intracranial recordings, and we make some predictions about the observed functional connectivity on the basis of the search information measure. And we actually do much, much better than people have in the past. So there's some predictive capacity from these very simple, exquisitely simple processes that are trying to capture some stylized version of communication in the brain. And there's more, and I would encourage you to read more about these. I'm running out of time, and I want to make sure I get to the end with some time for questions. Some based on like greedy routing through the network, utilizing multi-step paths, or how easy is it to get back on the shortest path if you were to diverge somehow. But this is a nice framework, we think, this communication dynamics framework. It generates better predictions of functional connectivity, computationally tractable, and it's flexible enough to incorporate almost any communication hypothesis that you have. And ultimately, we can use it to evaluate and compare those different hypotheses. So I've talked now about these biophysical and mechanistic. I've talked about the communication dynamics. A quick summary of this. So structural connectivity constrains function by shaping communication processes. We can use dynamical models that constrain structural connectivity to make predictions about function. Some of these models are complex, some of them are simple, but they both work and they're both suited for different kinds of questions. A really quick pitfalls and issues, and then I'll finish up. Right now, we're interpreting model fitness as a measure, how well it predicts functional connectivity as a measure of model fitness. It doesn't tell us anything about the correctness of a model. The biophysical model at the end of the day still retains much more of the flavor of neuroscience than the communication processes. So just because we can generate better predictions doesn't necessarily mean that those models are better or correct in any sense. All of these models use structural connectivity data to generate predictions. It's only as good as the data that comes in. And we've already heard at least one talk today illustrating some of the challenges, and there are many, some of the challenges associated with generating good structural connectivity maps. And then finally, there are also variables that confound the prediction. If we want to make a claim that functional connectivity is predicted by some communication process, we have to also take into account the other variables that could explain it equally well, like spatial relationships. Brain areas that are close to one another, they tend to be more connected for whatever reason. So we need to make sure that when we make that claim that structure is predicting function, it's not simply an effect of a spatial relationship. So I talked about a network science framework for representing structural and functional connectivity. I presented some empirical evidence that the two modalities are related to one another, and discussed some modeling approaches based on biophysics and stylized dynamics. And then I ended with some challenges and frontiers, and I'm happy to answer any questions.